Okay, in this video we're going to be looking at the G proteins, both the monomeric and the heterotrimeric G proteins. So in the previous video we've looked at the idea of protein modification by phosphorylation as a way of controlling protein activity. So effectively we had a protein that was phosphorylated by a kinase, leading to an active protein, which could then be dephosphorylated by a phosphatase, typically giving an inactive protein, although some proteins are inactive when they're phosphorylated, but typically we think of them as being activated by phosphorylation. Now, in a similar kind of reaction, we have activation and inactivation of a protein. However, it's not through phosphorylation, it's through the interaction of the protein with a nucleotide. So in this case, we have guanine nucleotides, and the protein, when bound to a guanine diphosphate, is inactive. And when bound to a guanine triphosphate, it's active. Okay? Now, there's an important part of this cycle of activation and inactivation is that activation occurs through nucleotide exchange and inactivation occurs through nucleotide hydrolysis. So activation occurs through exchanging a GDP for a GTP to give an active bound protein, and hydrolysis of this GTP leads to inactivation of the protein. Now, these proteins are referred to as G proteins, basically because they bind guanine nucleotides, and basically because they have an intrinsic GTPase. And the GTPase is the GTP being hydrolyzed to GDP. So there's a built-in off switch of these proteins through a weak GTPase. Now, there are two broad classes of G proteins that we're going to be discussing. The first is the heterotrimeric G proteins, and the second will be the monomeric G proteins. Okay. Now, here's a schematic of a heterotrimeric G protein taken, I think, from Albert's Molecular Biology of the Cell. And in this case here, you've got the three proteins, because it's a trimer, so it's a complex made of three proteins. We have an alpha, a beta, and a gamma subunit. The alpha subunit is the actual G protein part of this subunit, and here is shown the G protein bound to a GDP, therefore it's in the inactive state. And this is just showing the sort of some structural data of this complex and showing how the cartoon reflects the actual structure of the complex. Now, we've previously been discussing the activation and inactivation of these G proteins. Here is the same information but shown from the textbook. So this is from the Hancock textbook, and this is the G protein cycle applied to the heterotrimeric G proteins. So here's our alpha, beta, and gamma subunits bound to GDP in the inactive state. The GDP leaves, and the GTP is exchanged, and then this leads to the formation of the active form of the alpha subunit, and then the weak GTPase activity of this subunit can cause hydrolysis back to GDP. A typical example of a pathway that involves these heterotrimeric G proteins is the activation of adenyl cyclase. So here's our G protein coupled receptor bound to a tetramer of G proteins, with the alpha subunit being the stimulatory subunit, the actual GTPase. The ligand binding to the receptor causes exchange of GDP to be replaced by GTP, and then the activated alpha subunit with GTP can interact with adenyl cyclase, and therefore the adenyl cyclase becomes active, and its function is to convert ATP to cyclic AMP. Again, this information is well summarized in the Leninger textbook, so if you have a look at the chapter on biosignaling in Leninger, you'll see a nice pathway diagram describing what I've just explained to you. Now, we previously, in a lecture or two ago, looked at the activation of protein kinase A through allosteric interaction with ATP to give you the active catalytic subunits of protein kinase A. This is just showing some of the upstream events that lead to the activation of protein kinase A, which is a signal binding to a G protein coupled receptor, and then it's the heterotrimeric G proteins that are bound to the receptor that exchange and activate GTP, which then activate the adenyl cyclase, which is this big brown one shown here, and then the adenyl cyclase can then convert ATP to cyclic AMP, which can then activate protein kinase A. So this is a nice little example of activation of a second messenger through the heterotrimeric G protein complex. Bear in mind that not all G proteins activate adenyl cyclase. There are some G proteins that cause inhibition of adenyl cyclase, and also these heterotrimeric G proteins can interact with a range of other enzyme activities as well. And again, there's a nice table here showing a range of heterotrimeric G proteins here, referring to the alpha subunit. So the one we just looked at was the G alpha subunit being stimulatory, which is G alpha S, and this G alpha, the GS activates adenyl cyclase. There's a GI for an inhibitory subunit also, which inactivates adenyl cyclase. And as we've seen, we've looked at other, in the later lectures, we looked at a G-ALF, which is involved in odorant receptors in the nose. We looked at a GQ, which is involved in the activation of phospholipase C. And also when we looked at sensing light, we looked at transducin, or GT, which was involved in activating a cyclic GMP phosphodiesterase. So this table is just to remind you that there's a range of these heterotrimeric G proteins that have different effects on different enzyme activities. And again, this is a figure from the textbook, the Hancock textbook, which is just showing the same information. Here's the GS subunit of a heterotrimeric G protein, and the S is stimulatory for the adenyl cyclase, but also there can be a competing activity from an inhibitory G alpha subunit, which can lead to inhibition of the adenyl cyclase. So adenyl cyclase is one of the key activities that makes these cyclic AMP molecules. There's also a guanyl cyclase that makes, cyclic GMP molecules. These are the on switches for these second messengers. Just bear in mind that there are also cyclic nucleotide phosphodiesterases, commonly referred to as just phosphodiesterases, which break down cyclic G or AMP into G or AMP. So the cyclic AMP is converted into plain AMP. And there's a whole bunch of these phosphodiesterases that are involved in turning off signaling from these second messengers. So that was just a quick look at the heterotrimeric G proteins. Now we're just going to have a quick look at the small monomeric G proteins. So the key example of a small monomeric G protein will be RAS. So this is just showing the monomeric G protein here in an inactive state bound to GDP, and then there's a process that involves the exchange of the GDP and the uptake of GTP to give you the active monomeric G protein bound to GTP. In this cycle here is shown an empty state, and also in this cycle is shown a guanine nucleotide releasing protein, or a guanine exchange factor, which is involved in displacing the GDP so that GTP can then enter the pocket. And also we've been talking about how the G proteins have a weak GTPase activity. This is showing one of the helper proteins, a gap protein that's involved in speeding up the rate of this hydrolysis reaction, leading to an inactive state. And we'll discuss this more in a couple of slides' time. So this is just placing the RAS protein in context of a signaling pathway. So here we have a ligand binding to a receptor tyrosine kinase, which is then binding to an adapter protein such as GRB2, which can then bind to a guanine nucleotide releasing protein, or a GEF, a GEF, such as the SOS protein, which can then lead to the activation of a monomeric G protein such as RAS, which then leads to a cascade of kinase activities, which is our MAP kinase cascade, leading to an effect such as gene transcription. So RAS is our typical monomeric G protein that we discuss. RAS has been identified in many solid tumours, so its mutation is an important player in the formation of cancer. And surprisingly, a single nucleotide mutation leading to the replacement of GLI12 for any other amino acid can lead to a transforming overactive RAS protein, which can contribute to the formation of cancer. There's a little animation here just showing the RAS protein functioning as a molecular switch. So there are these loops or switch loops which exist in the RAS protein, which aren't highly structured, unlike the alpha helices. And here is a binding pocket for the nucleotide, whether it be GDP or GTP. And then as the nucleotide exchanges, then it changes the structure of the RAS protein. And there's a little animation here showing a switch in structure. So the structural change that occurs is driven by the presence or absence of this phosphate group within the binding pocket. So the protein starts off with a GTP changing, and then it just shows you when the GTP is exchanged for GDP, there's a switch, and then it's switching back to the GTP form. So this is just an animation showing how the presence or absence of this phosphate group leads to a change in structure of the protein, and various residues on the RAS are then exposed or hidden, leading to its change in activity. Now, there are 3 classes of helper proteins which modulate the activity of the RAS. The first one are these GEF proteins, these guanine exchange factors. And this is just a diagram from another textbook showing the guanine exchange factors helping the exchange of GDP for GTP to activate RAS. And again, this is showing another class of proteins, these GDI, these guanine nucleotide dissociation inhibitors, which function to maintain RAS in an inactive state bound to GDP. And then the other category that we mentioned briefly are the GAP proteins, the GTPase activating proteins. So the GAP proteins activate the GTPase of RAS, which hydrolyze GTP to GDP and therefore inactivate the monomeric G protein. So the GAP proteins lead to the inactivation of monomeric G proteins. The GAP proteins lead to activation of the GTPase activity. So under the influence of these GTPases, the rate at which a G protein can hydrolyze the GTP is increased many thousand-fold, meaning that it's the off switch. So the GAP proteins serve as an off switch so that the G protein can hydrolyze GTP and to GDP, leaving the protein in an inactive state. This is a quick diagram showing you that the RAS protein is post-translationally modified by binding to these long carbon chain molecules, these farnesyl and palmitoyl fatty acids, and these embed the RAS protein or attach the RAS protein to the inner leaflet of the membrane so that it can interact with membrane constituents. So assuming that we have the membrane-bound or membrane-attached RAS in an active form bound to GDP, what now? So RAF is one of the main effector proteins of RAS. So active RAS can activate or signal to the RAF kinase protein, and as you may recall, RAF kinase is the first step of the MAP kinase pathway. So we then have a series of events leading to gene activation and control of cellular processes. So RAF kinase is one of the central effector molecules of active RAS. RAF kinase is immediately downstream of RAS, and the activated form of RAS bound to GDP can activate the RAF molecule. A termination of RAF signaling occurs when RAF dissociates from RAS or when the GTP of RAS is hydrolyzed. Here's a dodgy diagram from one of the chemical companies showing the receptor tyrosine kinase being activated, leading to RAS activation and the MAP kinase activation. It's not really showing the GRB or the SOS in any detail, but it's a nice one that just shows RAS being activated and then the MAP kinase pathway being activated. This represents RAS activation in a very linear, easy to understand pathway, and that's how the book also talks about the RAS activation. But bear in mind that RAS is central to many pathways, so this is just one linear pathway within a network of pathways that may be active within a cell. So the RAS pathway cannot really be seen as a linear ordering of signal from one to another, but it's central sort of switching protein involved in various signaling pathways is how RAS is described in the textbook. So RAS can respond to many signals. Okay, I hope that was useful for you.